NRG1. NRG1 stands for proneuregulin 1, membrane-bound isoform. NRG1 is a direct ligand for ERBB3 and ERBB4 tyrosine kinase receptors. Concomitantly recruits ERBB1 and ERBB2 co-receptors, resulting in ligand-stimulated tyrosine phosphorylation and activation of ERBB receptors. The ERBB family of proteins contains 4 receptor tyrosine kinases, structurally related to the epidermal growth factor receptor, also known as EGFR. The multiple isoforms perform diverse functions, such as inducing growth and differentiation of epithelial, glial, neuronal, and skeletal muscle cells, inducing expression of acetylcholine receptor in synaptic vesicles during the formation of the neuromuscular junction, stimulating lobular-alveolar budding and milk production in the mammary gland and inducing differentiation of mammary tumor cells, stimulating Schwann cell proliferation, implication in the development of the myocardium, such as trabeculation of the developing heart. Isoform 10 may play a role in motor and sensory neuron development. The molecular functions for NRG1 are cytokine activity, ERBB3 class receptor binding, growth factor activity, protein tyrosine kinase activity activator, receptor binding, receptor tyrosine kinase binding, transcription cofactor activity, and transmembrane receptor protein tyrosine kinase activator activity. The biological processes for NRG1 are activation of transmembrane receptor protein tyrosine kinase activity, axon ensheathment, cardiac conduction system development, cardiac muscle cell differentiation, cardiac muscle cell myoblast differentiation, cell communication, cell migration, cell morphogenesis, cell proliferation, cellular protein complex disassembly, embryo development, endocardial cell differentiation, epidermal growth factor receptor signaling pathway, ERBB signaling pathway, Fc epsilon receptor signaling pathway, fibroblast growth factor receptor signaling pathway, glial cell fate commitment, innate immune response, intracellular signal transduction, locomotory behavior, mammary gland development, MAPK cascade, negative regulation of cardiac muscle cell apoptotic process, negative regulation of extrinsic apoptotic signaling pathway in absence of ligand, negative regulation of protein catabolic process, negative regulation of secretion, negative regulation of transcription, DNA templated, nervous system development, neural crest cell development, neuron fate commitment, neurotransmitter receptor metabolic process, neurotrophin TRK receptor signaling pathway, oligodendrocyte differentiation, peripheral nervous system development, phosphatidylinositol mediated signaling, positive regulation of calcineurin, also known as NFAT signal cascade, positive regulation of cardiac muscle cell differentiation, positive regulation of cardiac muscle cell proliferation, positive regulation of cell adhesion, positive regulation of cell growth, positive regulation of myelination, positive regulation of protein kinase B signaling, positive regulation of protein targeting to mitochondrion, positive regulation of protein tyrosine kinase activity, positive regulation of RAS protein signal transduction, positive regulation of striated muscle cell differentiation, regulation of protein heterodimerization activity, regulation of protein homodimerization activity, synapse assembly, transmembrane receptor protein tyrosine kinase signaling pathway, ventricular cardiac muscle cell differentiation, ventricular trabecular myocardium morphogenesis, and wound healing. The components for NRG1 are apical plasma membrane, axon, cytoplasm, extracellular region, extracellular space, integral component of plasma membrane, membrane, neuromuscular junction, and nucleus. NRG1 displayed evidence to pleiotrophy revealing associations with 4 or more endophenotypes related to schizophrenia. Data suggests a genetic association between SNPs and several genes, such as HTR2A, and response to LY2140023 treatment and schizophrenia. The protein encoded by this gene is a membrane glycoprotein that mediates cell-cell signaling and plays a crucial role in the growth and development of multiple organ systems. An extraordinary variety of different isoforms are produced from this gene through alternative promoter usage and splicing. These isoforms are expressed in a tissue-specific manner and differ significantly in their structure, and are classified as types 1, 2, 3, 4, 5, and 6. Dysregulation of this gene has been linked to diseases such as cancer, schizophrenia, and bipolar disorder. Several independent studies have identified neuregulin-1, NRG1, and its receptor, ERBB4, as important risk genes for schizophrenia. NRG1 and ERBB4 signaling controls the development of inhibitory circuitries in the mammalian cerebral cortex by cell autonomously regulating in the connectivity of specific GABA, also known as gamma-aminobutyric acid containing interneurons. NRG1 plays a role in increasing the genetic risk to positive symptoms of psychosis in a proportion of late-onset Alzheimer's disease families. NRG1 affects myocardial contractility and activates NOS in cardiomyocytes. Diseases related to NRG1 are schizophrenia, bipolar disorder, psychotic disorders, psychiatric status rating scales, schizophrenic psychology, Hirschsprung's disease, thyroid neoplasms, neuropsychological tests, short-term memory, mental disorders, reaction time, ocular motility disorders, startle reaction, cognition, cognition disorders, cardiovascular diseases, Alzheimer's disease, marijuana abuse, autistic disorder, major depressive disorder, cerebral dominance, schizotypal personality disorder, substance-induced psychosis, personality test, mental recall, intelligence test, periventricular leukomalacia, lung neoplasms, neurotic disorders, obstetric labor complications, adenocarcinoma, mental retardation, perceptual disorders, recurrence, psychological tests, psychomotor performance, tobacco use disorder, ventricular fibrillation, ventricular outflow obstruction, space perception, speech disorders, stomach neoplasms, psychological stress, task performance analysis, edema, psychological extroversion, functional laterality, inborn genetic diseases, type 2 diabetes mellitus, Beset syndrome, memory disorders, long-term memory, newborn infant diseases, psychology inhibition, congenital heart defects, amphetamine-related disorders, anorexia, asthma, atrophy, attention, carcinoma, non-small cell lung carcinoma, papillary carcinoma, carotid artery disease, pervasive child development disorders, chronic disease, sudden cardiac death, and depressive disorders. If you have any concerns about NRG1, contact a practitioner who understands epigenetics and nutrigenomics.